Hi, welcome back everyone for the second talk in today's session. The second talk will be delivered by Professor Madan Rao. Madan is a theoretical physicist and he is working at the NCBS Bangalore. He is also affiliated with the Simon Center for the Study of Living Machines. He has a wide variety of interests, applying physics principles to the understanding of active cellular biological systems. The title of his today's talk is The Physics of the Actomyosin Cytoskeleton in Cells and Tissues. Madan, please.
Yeah. Thanks, Saroj. So I should first thank the three Vs, Vijay, Vidya, and Vaishnavi, for organizing this interesting format for the meeting as a way to encourage your thirst for theory in biology. Though thirst, as all of you know, is a dangerous thing. Haisha has just led a very brilliant discussion on actomyosin networks and reconstituted systems. I, on the other hand, will talk about actomyosin in cells and tissues from a theoretical perspective. Now, I've been asked by Vijay to give a pedagogical talk, so that's what I'll do. And I ask the indulgence of the experts in the audience. Yeah, so this is just to describe the scope of the lecture. What I'll do is I'll discuss the many contexts in which actomyosin makes its appearance and influences shape, form, transport, rheology, and patterning in both cells shown here and in tissues. And so I'll be basically sort of focusing on these three messages that I'd like to convey to you. That actomyosin together form the molecular correlates of biological force in cells and tissues, predominantly. Actomyosin participates in both active force generation and force sensing, and this is important, that it does both these things. Finally, what I'd like you to see through this talk is that these components, these ingredients will naturally result in force patterning and excitability. And that's what I'd like you to take back from this lecture, is that the actomyosin system in living cells and tissues, quite distinct from the reconstituted systems that Aisha talked about, behaves often as an excitable medium and displays singular force patterns which come about as an interplay between active stresses, deformation and flows, turnover, and signaling. Okay, so I'll now sort of list the outline of this, of the presentation. I'll first, there's going to be a significant overlap with what Aisha talked about. And I'm sort of hoping that repetition might be useful to reinforce ideas. But I'll try to put a different flavor to each of the common ingredients that we have talked about. So I'll first talk about the physical properties of the building blocks, namely actin filaments, myosin motors, and the combination actomyosin units. I'll then go on to describe actomyosin as an active fluid, theoretical ideas regarding that. Then I'll talk about actomyosin as an active elastomer, as an elastic medium. And then we'll focus on the actomyosin meshwork with turnover components, which allows you to go from an elastic description to a fluid description. If time permits, I'll talk about coupled cytoskeletal elements, very similar to what Aisha talked in the last part of her lecture. And again, if time permits, I'll talk about actomyosin networks in tissues. Yeah, so let's start now with the first part, namely the single filament description of actin, of the elasticity of actin. And this is a very nice review article that's been written by Fred McIntosh and Rodersch. So actin filaments, as depicted here, is a long filamental structure built up of these monomers, and the typical dimensions are shown there. The elastic properties of actin, the mechanical properties of actin, can be described as a semi-flexible filament, which is inextensible. That is, the length or the filament does not change. Okay, that does not change significantly. And what we mean by semi-flexible filaments is that the mechanics or the elasticity, or sorry, the mechanics is governed by an interplay between entropy and bending elasticity. So indeed, being at the regime where energy and entropy effects are roughly comparable, they exhibit interesting linear and nonlinear elasticity and flow properties. Okay. So a theorist sort of describes these actin filaments, the elastic properties of the actin filaments, as this sort of worm-like chain model, where you sort of you hide the chemistry behind and describe it as a semi-flexible rod of thickness 2a, parameterized by an arc length s here. And as you go along the arc length, the tangent vector, which describes or characterizes the local curvature, is denoted by T sub s. Okay, the worm-like chain model is a description of the elasticity of this chain, this filament, and is given by this energy function, or the Hamiltonian. It says that as the curvature changes, as the local curvature changes as you move along the arc length, that costs energy. Okay, so this is called a curvature elasticity. The curvature is simply the rate at which the tangent vector changes as you move along the arc length. Okay, the modulus here is the curvature elastic modulus, okay, kappa, and for a cylinder like so, it can be written in as proportional to the Young's modulus multiplied by the fourth power of the radius of the cylinder. This is essentially the bending elastic modulus is essentially the Young's modulus times the moment of inertia tensor, the moment of inertia about the z-axis, about the long axis of the cylinder, and this is the formula. Typical numbers for the Young's modulus have been measured to be around 1 gigapascal. You can see that from this form of the energy that kappa has units of energy into length. Okay, and therefore one can define a length scale called the persistence length, which was referred to by Aisha, as being the ratio of kappa to kT, the thermal energy. Okay, and this typically for actin filaments is around 17 microns. Okay. Now the contour length of actin, of course, varies, and it takes a huge range of values within the cell. And it could range anywhere between, where the upper limit is around 20 microns, to the lower limit could be around 250 nanometers. This might appear surprising, the lower limit, but has been recently been measured through a variety of techniques. In fact, in our group here, where in Jitu Mayer's lab, where they have used TIFR FCS to measure the length scale of these tiny actin filaments close to the cell surface, speckled single molecule imaging, and EM. Okay, and so there's a variety of techniques. It seems that the actin filaments can have a range of lengths going from 250 nanometers to 20 microns. In any case, you can see the typical lengths of actin filaments are smaller than the persistence length. Okay. Now, because of thermal fluctuations, which is related to entropy, the semi-flexible polymer is crumpled. Okay, crumpled at very long length scales, with the tangent-tangent correlation function decaying exponentially as so. So the tangent at 2 points on the semi-flexible polymer decays exponentially over a length scale which is equal to the same persistence length. And so this would it would appear that from this formula that thermal effects will only manifest at length scales much larger than the persistence length. And the persistence length, I'd like to remind you, is 17 microns. Okay. However, surprisingly, that's not true. And even for length scales smaller than, in fact, quite a significantly smaller than, the persistence length, thermal effects are very crucial. And the way to understand that is the following: that if I take up an actin filament and sort of tether it, this is a thought experiment, tether it at one end here and allow the other end to fluctuate, then you find that because of these bending fluctuations, okay, which are quite significant, the length, the projected length of the actin filament gets shorter. Okay, and so there are both transverse fluctuations. Sorry, this mouse is not moving very well. There are both transverse fluctuations because of bending fluctuations, and also as a consequence of the shortening, there are also longitudinal fluctuations. Okay, and both these fluctuations are soft or very easy to excite. Okay, and indeed, when you apply attention to iron out or straighten out the polymer or iron out these bending fluctuations, then you'll increase the length. Okay, and therefore the tension is actually conjugate to this excess length that you pull out. Okay, so it goes to reduce the entropy, and therefore you're doing work against entropy. Okay, so now to get an estimate of the amount of both the transverse and longitudinal fluctuations, we'll have to do a little calculation. And the calculation is easiest done in a certain representation called the Monge representation, which is valid when the length of the polymer is smaller than the persistence length. In such cases, there are only tiny fluctuations around the straight line along the x-axis. And therefore, I can, instead of talking about arc length, I can write down the transverse displacement of the polymer as a function of x itself. Okay, this sort of simplifies the calculation and allows an easy estimation. So in this sort of representation, the bending energy is like so. This is again the curve, the local curvature square, integrated over the length of the polymer. Okay, multiplied by the bending modulus that bending elastic modulus that I defined earlier. Okay, a simple Fourier transform, and this is the definition of the Fourier transform. A simple Fourier transform will relate this real space energy into this momentum space representation. Okay, it's sort of nicer to look at it this way because this is just a set of independent modes for which you can use equipartition of energy to quickly estimate what is the typical fluctuations, both in transverse and longitudinal fluctuations. Okay. Now one can ask, what is the excess length that is stored in these bending fluctuations? Okay, and the excess length that is stored in the bending fluctuations is given by this formula. This is the typical excursions that the polymer makes transverse to the x-axis. Okay, and therefore the first term here is the length of the polymer, and this, when I minus it from 1, is minus the projected length. Okay, so this is this excess length that is stored in the due to the out of plane fluctuations of the polymer. Okay, and since one is in the Monge representation, one can readily expand the square root, okay, and keep only the first term, and you get this as the formula. So it turns out that the extension delta L, okay, the longitudinal extension delta L is related to this quantity here. Okay. Now, as I said, the force, the tension tau is conjugate to this delta L. Okay, because you're pulling out the excess length stored in the out of plane, out of axis transverse fluctuations. Okay, so the resulting new Hamiltonian that you would like to write down would be the old one, namely the bending elasticity plus a force multiplied by the extension delta L, which we just shown to be this form. Okay, so this is now the Hamiltonian, which describes both the bending elasticity and the response to an external force. Okay. Now, again, as before, it's easy to take via this Fourier transform method. It is easy to write this as a sum of independent modes. Then I use equipartition, which does I distribute the energy of each mode as half kT, and then I integrate over all values of the wave vector. Okay, this is a standard technique that is used in physics, and this immediately gives you what is the typical transverse fluctuations and the typical longitudinal fluctuations. Okay, and this is the formula that you get in the limit that this force goes to 0. Okay, the transverse fluctuations is inversely related to the persistence length and directly related to the cube of the length of the polymer. The longitudinal fluctuations is again inversely related to the persistence length, but related to the square of the length of the polymer. This is easy to see from dimensional analysis. U square has the dimensions of length square. Clearly, the transverse fluctuations are going to be large when the temperature is large, or when the bending elasticity is small, and therefore it should be inversely proportional to LP. Okay, because remember the formula for LP is kappa by KT. Okay? And the same argument holds for the longitudinal fluctuations. Both should be inversely related to LP. And then you bung in as many powers of L upstairs, because that's the only other length scale in the problem, to give you the dimensions of a square length here and a linear length there. Okay, so this immediately gives you this dependence. Okay, so what does this show you? What this shows you is that delta L, okay, quite surprisingly, delta L is quite large. The longitudinal fluctuations are indeed larger than the transverse fluctuations. Okay, and it's easier to it's very easy to excite the longitudinal fluctuations. Okay, so therefore, this will be significant even for length scales which are small compared to LP. Okay, yeah. Now, so this was the calculation that we did when the force that is pulling out this excess length goes to 0. After all, we want to calculate what is the force extension relationship, namely how does the polymer respond to mechanical forces. Okay, so we need to ask what is the physics when you have a finite tension tau. Okay. When you have a finite tension tau, you immediately find that there's a new length scale in the problem. The old length scale was LP, the persistence length, which was given by the ratio of kappa, the bending modulus, elasticity to KT, elasticity to entropy. Now you have a new length scale coming in, which is the ratio of the bending modulus to the force that you apply. Okay, so, and this is very similar to the length scale that Pierre must have talked about in yesterday's talk. Okay, now all the discussion that we have had so far is valid when thermal fluctuations are high. That is when the length of the polymer is smaller than LT, the length due to the tension. Okay. When the length of the polymer is much larger than LT, then thermal fluctuations are not that important. Okay, not very important. Okay. So now in order to calculate the force extension curve, I first need to measure how does tau vary with the extension. Okay, that's what the force extension curve means, right? So I apply a given tau and I ask what is the apply a given force tau and ask what is the extension of the polymer, the semi-flexible polymer, and clearly the extension is just related to this quantity. Okay, is the difference in delta L at tau equals 0 minus at the fixed value of tau. The strain is simply the change in the length delta L divided by the original length. Okay. So if I can calculate delta L, which I can do from the previous kind of calculations that I've shown here, and I calculate delta L0, which I've already done here. Okay, then I will be able to calculate what the strain is, and I can then plot how does tau vary with, or how does the extension, the strain vary with tau. Okay, so here's the graph that you get. as a result of, it's a numerically determined graph from the formula that can be easily derived by the procedure I told you about. And this tells you that the dimensionless force here, okay, related to tau, versus the extension, the strain epsilon. Okay. What it shows is that for length scales much smaller than the tension length scale, the form is linear, okay, which is the standard form that you get for a thermally dominated regime for the polymer, because the elasticity is thermally dominated. Okay, and at large values of the length compared to LT, you go over to this nonlinear regime. Okay, this is the linear to nonlinear stiffening that Haisha talked about in her lecture. This is a function of the strain epsilon. Okay, this is for extension. Okay, this curve here is for extension, and this curve here is for compression. Okay, and I'll come to the compression part later. Okay. So again, I want to repeat that the force extension curve for these semi-flexible polymers has an initial form which is linear, and then has a nonlinear strain stiffening at large values of extension of strain. Okay. Okay. Now, the elasticity that we are just describing here is, of course, significantly determined by thermal fluctuations. But of course, after all, if you pull the polymer and iron out all the thermal fluctuations, enthalpic forces will also come into play, namely forces coming from interaction between the elements that build the polymer. Okay, so the question is, when does this enthalpic elasticity start dominating? And what one can show is that when the length of the polymer, when this formula is satisfied, namely the length of the polymer cube is larger than the a square, which is the thickness, the radius of the semi-flexible rod multiplied by the persistence length, then entropic features are dominant. So large enough L, entropy dominates. Whereas for smaller L, small compared to this quantity here, enthalpic forces dominate, and that governs the elasticity. Okay. Yeah. So now, so far I've been discussing how does the polymer respond to extension. You could ask then, how does it respond to compression? And as Haisha talked about, these polymers such as actin, they don't, they buckle very immediately when you apply a compressive load. Okay, so they can't sustain a lot of compression. And if it was a free actin polymer here of length L, then the sort of critical force required to buckle this actin filament will go as kappa divided by L square, which is a standard Euler critical force. Okay. In the cell, of course, you don't get filaments, isolated filaments on their own. They're embedded in a meshwork that comprises the rest of the cytoskeleton of the cell. And it turns out that because of the elastic meshwork that surrounds this actin filament, the buckling, the critical buckling force is higher. Okay, it can sustain buckling. It can sustain compression to a certain extent. The critical buckling force goes from being kappa by L square to 2 kappa by lambda square, where lambda is given by this length. The physics of this is that in order for this to buckle, it needs to distort its shape. This distortion of the shape will then compress the elastic medium here. That compression will cost an energy which is related to the compression stiffness of this of the meshwork. Okay, and so so instead of buckling over a length scale which is equal to this length of the polymer or half the length of the polymer, you actually start buckling at much smaller wavelengths. Okay, and so this has two effects. One is that it makes the most dominant wave vector or wavelength to be much smaller than this, the most unstable wavelength to be much smaller than the Euler load, but it also makes the critical Euler load much larger. Okay, so the bottom line now here is that actin filaments have an asymmetry in the response between extension and compression. They are easy to compress because they buckle. Okay, so far we've been talking about the elasticity of of actin. I'll now talk about actin nucleation, growth, and turnover, the dynamics. Okay, so let's just discuss some of the features of actin dynamics that will be relevant to the cell. One feature is that that actin has to be recruited; it gets nucleated, and the nucleation and growth of actin. The nucleation and growth of actin is governed by signaling processes. Okay, and this is very important to realize. And this is what makes a huge difference between the phenomena or the physics of what is happening in the cell compared to what happens in vitro context. And as Haisha was saying, that one of the aspects that I'd like to highlight here is the mechanochemistry and the strong sense of regulation that both actin and myosin have in the context of the cell. Okay, so the nucleation and growth of linear filaments and branched filaments, and actin appears both as linear filaments and as branched filaments. They happen because of some nucleating factors. They're called Rho GTPase, and how exactly Rho responds to various cues is an ongoing set of research. It responds to both chemical and mechanical cues in ways that we're in very complex ways that one's trying to understand. Okay, so the bottom line here again is that chemical Rho signaling is responsible for the recruitment, local recruitment and growth of actin. Okay, actin filaments, as Haisha talked about, have a structural polarity, and this leads to an asymmetry in the polymerization and depolymerization. And so the polymerization in the polymerization rate in the barbed end and the pointed end are different. Okay, and this gives rise to this phenomena called treadmilling, as shown here. Okay, there's a time series of labeled actin, which shows the phenomenon of treadmilling. Okay, and so polarity and treadmilling are consequential to the following feature: that if I put a wall here, then because of treadmilling or because of polymerization, there will be a force that the polymerizing actin filament applies to the wall, and that force will be proportional to the orientation. Okay, so that's the take-home message here, that the force of the polymerizing filament here is proportional to its orientation. Okay. A further feature of actin filaments that needs to be borne in mind is that actin filaments typically bundle, and while single actin filaments are polar, bundles of actin filaments need not be polar because they can be aligned in opposite polarity fashion. Okay, so they can, as I said, they can form branched bundles like so, which are regulated by this protein called R23. They can also form these linear bundles, which Haisha talked about, due to fascin and alpha actin. Okay, and in doing so, collections of these actin filaments, both branched and linear filaments, can form two-dimensional and three-dimensional meshwork. This gives rise to a mesh size. Again, so you'll see that as we go on, there'll be more and more length scales that come into the play, and each length scale brings in their own special physics features. Okay, here the new length scale that comes about is the length, the size of the mesh work. Okay, which is generated as a because of the bundling and cross-linking of these massive filamentous mesh of actin. Okay, so these, as I said, these two-dimensional and three-dimensional meshwork of filaments, of filamentary bundles, are typically cross-linked, and this sets a length scale which is given, which I'll call xi, which depends on the density, of course, of the cross-linkers that bind to actin. Okay. Yeah. For these meshworks with crosslinkers, as Haisha was talking about, is the crosslinkers that determine the mechanical response more than single filament properties. Okay, and this is an important feature to be borne in mind, stressed very much by Haisha. Indeed, what she also talked about is that it is the presence and the occurrence of these crosslinkers that are important for maintaining contractile stresses in this actin meshwork. Okay. Yeah. The other feature that I'd like to discuss is that once these, this was a way to build up meshes, to build up bundles and to build up meshes. But of course, in the cell, there are enzymes such as cofilin, which is again referred to by Aisha, which serves as severing agents. So they break and cut, sever the filaments at designated points on the filaments in an energy-dependent manner. And both the breakage of the filaments due to cofilin and the unbinding of cross-linkers, so the turnover or the relaxation of stresses happening because of the unbinding and breaking of happens in a stress-dependent manner. Okay, and here you can see that actin and actomyosin serve now as force sensors because they can regulate their internal stresses by relaxation due to cross-linker unbinding and severing due to cofilin. Okay, now talk about so far the story was on actin. Now I'll talk about myosin very quickly. Some single molecular motor characteristics of myosin that one needs to bear in mind. One is, of course, that is ATP dependent conformational switches that allows it to walk literally on these actin filaments. There are many different kinds of myosin. Haisha mainly talked about myosin 2, which forms these bundles, okay, that she talked about, which are due to electrostatic interactions between the tails, form bundles of around 30 to 50 myosin heads. But there are also myosin 1 and many other different kinds of species of myosin. And indeed, they have different cellular localizations. Myosin 1, for instance, is mainly localized in the plasma membrane. And possibly in other membranes, but I don't know. But a lot of mainly in the plasma membrane. Myosin 2, on the other hand, is localized in the actin cortex, just beneath the plasma membrane, and in stress fibers. And it's this sort of spatial distribution of these different kinds of myosin, regulated by a variety of enzymes and regulators, that gives rise to force patterning in cells. Okay, myosin, just as actin was recruited by Rho and ROC signaling, similarly, myosin is also recruited by Rho and ROC signaling. So it's brought to a particular place in the cell because of ROC signaling, and thereupon it forms myosin 2 forms bundles, like I just mentioned, and subsequently, it sort of binds and it binds to actin where it does its power stroke activity. Okay, so this is a typical ATPase cycle where myosin mini filaments bind to actin. They bind and so this is the binding part of the cycle. This is the unbinding part of the cycle, and in between is a power stroke. And each of these binding, unbinding, and power stroke involve chemical cycles, which involves ATP, and it's this entire cycle that gives rise to actomyosin contractility. Okay, so now I'll talk about so I've now talked about actin and myosin separately, and we'll now bring them together to discuss the active currents and active stresses generated by this actomyosin unit. Okay, this is a picture taken from, in fact, Haisha's and Paluk's review article, which talks about the different ways in which myosin mini filaments can indeed engage with actin. Okay, the first one is when myosin motors engage with work on actin filaments towards the plus end to move actin filaments around if myosin is held. Okay, this gives rise to a current, which is again proportional to the orientation of the filament. In configurations such as these, the myosin mini filaments draw in or draw out actin filaments to give rise to a contraction or expansion, and this gives rise to local contractile stresses and local extensile stresses that again Aisha talked about. But remarkably, okay, in the cell, what predominates are these contractile stresses. And as Aisha was talking about, this was the subject of much interest and discussion in the community, whereupon it's been now realized that there are at least two different mechanisms by which the contractile activity of actomyosin dominates. One is because of this interesting single filament mechanical property that I talked about, namely the asymmetry in its mechanical response to compression versus tension. Because of compression, buckling here biases the system towards contractility, okay? And that's why this is what predominates in the stress relaxation of this actomyosin network. Okay, so you can see that locally, if I have this, and you have crosslinkers here which hold the actin filaments together, myosin mini filaments attach to the different actin filaments like so. This contraction process will sort of buckle the actin filament here, drawing in these things together. Okay, and this is why contraction dominates. The other reason, again, as Aisha talked about, was because of polarity salting, namely myosin, which moves towards the myosin head, which moves towards the plus end, can draw in filaments, actin filaments, in this kind of aster-like fashion. And again, it draws in, and therefore it gives rise to contractile flows. Both these features, namely polarity sorting due to myosin and active stresses coming because of because of this buckling, gives rise to contractile active stresses and contractile flows. Now we'll see that just as we had the current being proportional to the orientation, as we saw here and in the previous slide, the active stresses, the contractile active stresses that are obtained as a result of these two features also are proportional to the orientation. Okay? And the way to see this is to realize that these active forces are indeed internal forces. The force that the actin applies on the filament is the same as the filament applies on the actin. The whole system is is a momentum conserving system. It's an internal force. There are no external forces, and therefore the integrated force over a certain small coarse-grained region should be equal to 0. This immediately means that the force can be written as a divergence of a stress. Okay, and the idea now, and this means that there is no monopole force contribution. That indeed this relationship would, because of the absence of any monopole contribution, the force can be represented as a distribution of dipoles or higher moments. Okay? So because they are internal forces, I'm going to represent the forces and torques that are described in these two slides, okay, as a collection of force dipoles or torque dipoles. Okay, there's a simple way to estimate what is a typical form for the stresses of that myosin, that this actomyosin complex or unit applies in this locally. And without going through the details here, one can just show that the stress is actually proportional to the orientation of this filament. Okay. Q here is the orientation, which is built up from so N. Okay, yeah. Q is sort of the orientation of the rod, namely the nematic orientation of this rod. Okay, so the local stress that you obtain by resolving the individual forces of the force dipole around a particular point here, it can be written as a divergence of a local stress, and the stress is proportional to this orientation. Okay, so both the currents generated by the contractile flows by myosin and the stresses, the contractile stresses coming from actin and myosin are proportional to the current, are proportional to the orientation. Sorry. Oops, sorry. Okay. Okay. Now, so far I've been just discussing the orientation of a single unit here. Okay. So this single unit shown here more elaborately, I'm just sort of coarse graining it into this schematic here. Okay. And the orientation is just given by the single rod, by the single line here. Now, if I have a collection of such objects in the medium, the question is, how do I describe them, the orientation? And I just want to relate some nomenclature here. If I'm going to represent the coarse-grained or collective orientation of these units such as these, by the nematic order Q or its polar order N. Okay, and I'm going to describe it as follows. Each of these arrows display the filament orientation. Okay, and so if a collection of units that I described earlier point in this line or sort of orient in this configuration, I'll call them isotropic because they point every which way. If they orient predominantly along this direction, then this would be called a polar orientation because all the plus ends are on one side and minus ends are on the other. The polar orientation. If they are oriented in this particular way, so the plus and minus ends are along these directions, but the plus, one end is pointing this way and the other end is pointing that way. But they are all predominantly arranged in this particular direction. Okay, so there is no fore aft. There is fore aft symmetry. That is, this region is very similar to that region. This kind of orientation is called a nematic orientation because it doesn't have a polarity, but still it has a certain direction. Okay, a nematic direction. And I'll also call this as a nematic direction because here you have bundles of actin which are coming together in an apolar fashion, but they're roughly oriented in a nematic direction along this. Okay? This nematic orientation is sort of described by an orientation tensor Q, which is built from these little n vectors which describe the polar orientation of each of these filaments. Okay, so I'll pause here and by just saying the following, that the forces that one that gradients of sorry. Okay. Okay, I'll pause here to take some questions. Okay, and if you have any questions on this part of the lecture, then I'll be happy to take that.
There are right now a couple of questions. Sujit, you want to go ahead and ask the question?
Yeah, so I had questions in your very initial few slides actually, where you showed the extension. Yeah, yeah. The next one.
Yeah.
Yeah. So here your calculations are your calculations done for 2D in two
Uh,
dimensions?
no, so the drawing is of course for 2D, but you can do the, so U could be a vector. So in 3 dimensions, U has 2 components.
Yeah. Oh,
Yeah.
I
So,
see. So how different will it be in 3D?
so
All
this is
these.
the 3D calculation. I forgot to put the vectors everywhere. So sorry.
Oh, I see. So these are in the generic case, you're saying.
This is in the 3D cal. Yeah, yeah, yeah. So
Okay.
this number 6, for instance, comes from the 3D.
Yeah, I see. Okay, thank you.
Next question from Vaibhav. Vaibhav, do you want to go ahead and ask?
Madan, yeah, in this particular slide, when the medium is there, it appears that there is no dependence on length, and I didn't get that because, in the bucket case, when the medium is there, the lambda doesn't seem to depend on the length, and I didn't get that because once the medium disappears, you have to get into the other limit where everything was dependent on length, right?
Sure. So the point is that B here is the elastic modulus of the medium here, right?
Yeah.
So the elastic modulus of the medium depends on the crosslinker density.
Yeah.
So as you reduce the crosslinker density, so remember you'll only get a non-zero value of the compressive modulus when the crosslinker density exceeds the rigidity percolation limit. Okay, so when the crosslinker density is smaller than the rigidity percolation, then B goes to 0, and you then go to that.
Okay, so the equations themselves, they basically change. When B goes to 0, you're basically getting something else altogether. You're getting the transition.
Right. Because you would imagine that there's a phase transition.
Okay.
Right, because you're going from, if I start from here, then I can by reducing the density of crosslinkers, I can reduce B. And so I can go to this picture on the left-hand side by reducing B beyond the rigidity population.
Okay, and then lambda basically becomes L. That's the problem. That's the issue. What happens there? Something like that.
Exactly. Yeah. Right,
Okay,
because
okay.
lambda will keep increasing. Right, as you reduce B, lambda keeps increasing.
Yeah, and then somewhere along the line, the length basically appears. Okay, that's fine. Thanks.
Yeah. Sorry?
No, then somewhere along the line, even though B is going to 0, in principle in that equation, B goes to 0, lambda goes to infinity, but something else is happening basically, as you said, when there's a phase transition, and that's where the length basically appears. Okay, thanks.
Okay, so there is another question from Garima. Garima, go ahead, please.
Hello. So in the last part, you talked about the elasticity and orientation of filaments. So what about motors? Are they point objects, or do they also have some orientation or specific elasticity?
So right. So in the motors, yeah. So in many, so of course this is it's a big molecule, and if you want to sort of represent, you could, for instance, and this is one of the popular ways of representing myosin, is to treat it as a bead and spring model with the heads attached to. So you could treat this backbone, this backbone here as a rod where you have springs attached to these heads. Okay, and that's one way in which numerical simulations model myosin mini filaments. Yeah?
Oh, but do they follow linear constitutive relations? Like, is it confirmed or?
So I don't believe that I mean, these are all cartoons for it's a complicated molecule, and these are cartoons for the elastic behavior of that molecule itself. I mean, in what I'm going to talk about later, these will be treated as point objects.
Oh, okay, sure.
Okay. But your point is well taken, that how good are what agent, if I want to represent this by a microscopic agent-based model, are there good microscopic descriptions of this? And I believe not very. Even what we have done a bunch of agent-based simulations, and as I said, that's how we describe myosin. And and so the elastic moduli of that is. I'm not sure how well this.
Sure.
Yeah.
Okay. Thanks.
But these the important thing is that these filaments can be quite large, and so they quite flexible.
Yeah. So, but they will affect the orientation of actin filaments, I guess.
Yeah.
Okay. Yeah. Thank you.
Okay. So next question from Bharat. Please go ahead.
Nice talk, sir. I wanted to know if the transverse modes are actually independent of these longitudinal modes.
The transfers. So the Hamiltonian, as I said, is given by these transverse fluctuations, right?
Yes.
Right? And the moment. So the interesting thing here is because of this transverse fluctuations and because of inextensibility, the transverse fluctuations will immediately give rise to longitudinal.
Okay.
They're related, right? Imagine taking and it's actually shown nicely in this picture here. Because of these transverse fluctuations, you can see that this length here, this projected length, right, is smaller compared to if I apply a tension, which will draw out the transverse fluctuation.
Right. Okay.
So when I don't apply any tension, then you should think of this transverse fluctuations going like so, and longitudinal guy is also doing that.
Okay.
And since both the transverse fluctuations and the longitudinal fluctuations have their origin in these bending modes, they are related.
Okay. Thank you.
Okay, so there are two more questions. Shivani, go ahead, please.
Sir,
I can't hear that.
Okay, so the audio seems to be not clear.
Yeah. Of the representation of the the representation of the
Okay, so maybe I read out this question.
The representation of the orientation diagram that you gave, was it of actin, myosin, or the whole system?
Say that again, sorry.
Yes.
So, Madan, I read out this question. The representative of orientation is of actin, myosin, or the whole system?
The representation here is of the unit of actin and myosin.
So, yeah. There is another question from Sujit. Please go ahead. Okay.
Yeah, so I was just asking, could you please explain this expression of orientation tensor a bit? I mean, just how did you write it? Like n tensor and I mean, what is the motivation?
Okay. So imagine that. Now let me see here. Okay. So imagine that you're talking about a rod, right?
Yeah.
So suppose maybe this one. So this is clear, right? That the orientation here is given by this. Suppose I describe each of these arrows as an orientation vector n. Right? Then the local average of this collection of things will have some non-zero value of the average mean n.
Yeah.
Okay. Now, if you have half the arrows pointing that way and half the arrows pointing this way,
then zero.
Yeah. And of course, the average n is zero.
Yeah.
Right? However, this is not zero. So this is ni. So this tensor is qij. It's equal to ni nj.
Yeah, correct.
And so that will be non-zero here.
I see.
I'm going to take an average of the entire quantity. I'm not taking an average of the individual guys, but I'm going to take the average of the entire. Okay? And that will be non-zero. Okay. So this is a standard. This is a standard orientation tensor that is defined in liquid crystal physics, and yeah.
Okay.
And you're just separating out the baseless part. So this is a purely anisotropic.
I see.
Madan, there are more questions, but maybe would you like to take them now, or should we move on and get back later?
Yeah, maybe, unless they are very important.
So maybe we can get back to those questions later.
Okay.
And move on later.
So I guess so far what I've done is I've described the elastic properties of actin filament, the properties of myosin, the properties, the local elastic or mechanical properties of actomyosin, how it gives rise to both currents and stresses, and both the current and both the active current and active stresses are proportional to the orientation. Okay, so that's the take-home message here, and the orientation is defined by this tensor Q. Okay, this orientation tensor Q. Okay, so now let's talk about taking a collection of these polar actin filaments and myosin 2 motors. A suspension of these actomyosin units and place them in water. So fluid suspension of actomyosin filaments and myosin 2 motors. Okay, so we'll be looking at first at the fluid description, and the fluid description would be valid when the turnover of the crosslinkers is much faster than the stress relaxation times. Okay, it will be a fluid in the limit. So that's an important ingredient. Fast turnover of crosslinkers is when the collection, this fluid suspension of actomyosin filaments would behave like a fluid. Okay. It also behaves like a fluid in the limit, in a very dilute or semi-dilute limit. That is, as long as there's no entanglement. Okay. So in these limits of small filament length, low concentration of filaments, and fast turnover of crosslinkers or low density of crosslinkers, it will behave like a fluid. Okay. Now, to describe both the patterning and the mechanical properties of such a fluid of active elements, people have used agent-based simulations of actin polymers and myosin motors using bead spring models and motor activity that I just described, that I just replied to one of the questions that was there earlier. Okay. However, the orientation patterns, okay, that one sees in such agent-based simulations don't seem to reflect what you see in cells and tissues. And indeed, as Haisha talked about, in vitro experiments, the orientation patterns and that you see in these in vitro experiments too don't resemble. Sorry. Don't resemble what you see in vivo systems. Okay, typically the orientation patterns that you see in vitro context or in these agent-based simulations are jammed. They're not dynamic, and they are very, very far from what you see in vivo. Okay, and indeed, what I want to highlight in this part of the lecture is that we need to include turnover to get turnover of the components of actin and motors in order to get dynamic non-equilibrium steady states as seen in vivo and in a few in vitro experiments. Not many, but a few in vitro experiments. Okay. So what I'll focus on is a more sort of coarse-grained hydrodynamic description of this active actomyosin fluid. Okay, it's coarse-grained over length scales, which are going to be larger than the filament size. We're going to say that the filament size here as I described the length scales in my first or second slide, that the filament sizes are much smaller than the persistence length. Okay, and so in my hydrodynamic formalism, I'm going to take this as a typical sort of representation. Okay, in such a hydrodynamic formalism, I need to describe, of course, what my coarse-grained variables are. The coarse-grained variables that we are going to use are going to be the concentration of actin filaments given by C. Okay, the orientation of the actin filaments either given by a polar vector N or the orientation tensor Q. Okay, the bound myosin concentration. So myosin can bind and unbind onto the actin filaments. The bound myosin concentration is going to be rho B. The unbound myosin concentration, that is myosin not sitting on actin filaments, not bound to actin filaments, is going to be rho U. And finally, this entire filament, actomyosin filaments, are embedded in a Stokexian fluid, a high viscosity fluid. And so when these, one of the coarse-grained hydrodynamic variables will be the local hydrodynamic velocity of the Stokexian fluid called V. Okay, I'll need to describe locally what the active stress is, which comes from the binding of myosin onto actin. Okay, and the local stress, as we discussed earlier, is going to be a function of the concentration of the coarse-grained stress here. Is going to be a function of the local concentration of filaments, the orientation tensor of the orientation of the filaments, and the number of myosin motors bound to that to the sets, to the local collection of filaments. Okay, so this is a coarse-grained active stress. Okay, so this is the full description, and of course, this is many variables. Very difficult to describe it, but it has been done. Okay, but there's a simpler description that one can have, and which I'm going to call a scalar theory, which is going to club the concentration of filaments, the orientation of filaments, and the bound myosin density into one single variable, which we'll call rho. Rho is like the local density of the active agents. Okay, so now having clubbed these three hydrodynamic variables into one hydrodynamic variable, namely rho. The other hydrodynamic variable is the hydrodynamic velocity, V. It's now much simpler to write down the dynamics of rho and V in the scalar description. Okay? And typically, there are many variants of this kind of theory, and this is typically what the form is. The local density of these active agents, the time dependence of that is given, has an advective part that is advected by the local velocity. And there's a possible turnover. The turnover, which is a function of rho, can be written as a rate of binding minus the rate of unbinding. Okay, so this is a typical form, phenomenologically written form, written for this active density of active agents. The equations that describe the hydrodynamic velocity, as I said, is in this overdamped Stokes limit, is the force balance equation, and it balances the viscous force given by some friction coefficient multiplied by the velocity. Okay, balances that with the dissipative viscous force coming from the viscosity multiplied by the derivative of the strain rate, plus an active force, plus a possible chemical potential due to this density field. Okay, so this is just a balance of forces. I'm balancing the dissipative forces to the active force and the chemical potential. As in the scalar theory, we have sort of gotten rid of the orientation field. And so what is left of the activity is simply written in terms of rho. And therefore, the active stress is going to be a function of that rho. Okay, and this is a typical form one could take. This shows that for low values of rho, it increases with rho. And then for larger values of rho, it saturates. Okay, so it's a typical form which increases linearly and then saturates. Okay, for contractile, since actin, as we discussed, provides local contractile stresses, that fixes the sign of this parameter zeta1. Zeta1 turns out to be negative, and it's with this sign convention that the stress that one derives here is a contractile stress. It draws in material. That is, local enhancement of rho will draw more rho from its neighbors. Okay. So these are the set of equations. Simple enough. Equation for the active density of active agents, equation for the local velocities, given by the Stokes force balance conditions, and a constituted relationship between the active stress and the local myosin or local active density. Okay. Okay. Now, okay. So this was a description where I didn't. This was a scalar description where I didn't bother about the orientation. When I have an orientation, the equations are slightly more complicated, and I write down now equations for the concentration of the filament, orientation of the filament, concentration of bound myosin, and so on. Okay, and this, I'm just going to flash this but this can be done and has been done. And the equations, as I said, are more complicated, but in terms of words, they're easy to stress. So I'm going to, so this now here is the equation for the dynamics of the myosin. The equation for the dynamics of the myosin has a standard advection term. Bound myosin will move along with the filament, okay, and will move along with the fluid surrounding it, okay. And so that's the advection term with a current given by this formula, okay. And it has a turnover, the fact that myosin can unbind and bind, okay. So this is the binding and this is the unbinding, okay. So again, the dynamics of the density of myosin is simple. It's given by an advection due to the local velocities that come because of active advection, and this turnover coming from unbinding and binding of myosin. That's the dynamics for the myosin. Now I have to give you dynamics for the concentration of the filaments and then the orientation of the filaments. The concentration of the filaments is also given by a local advection because this is a concentration. Okay, and so the local filament concentration is also given by an advection here, but this is now the filament advection. And the filament advection is given by a filament diffusion term. The current is given by filament diffusion term, and these active contributions coming from the filament orientation. Okay, yeah, yeah, and finally the equation for the orientation is slightly more complicated, oops, sorry. Is slightly more complicated and is written as a sort of active version of traditional dynamics of orientation in liquid crystals. Okay, I'll just state it that way, and but this just says that the time derivative of the local orientation is given by some nonlinear convection terms, which arise from these active stresses. And on the right-hand side are terms which suggest that filaments like to locally align themselves because of crosslinkers and so on. Okay, and because of myosin, they like to form contractile regions like this. Okay, remember this is what I said about polarity sorting. Bound myosin locally likes to form these contractile astral-like regions. Okay, so this part of the equation, okay, it relates to this. This part of the equation relates to the fact that the filaments like to orient parallel to each other because of crosslinkers, and this part of the equation says that because of polarity sorting, filaments like to form these local asters. Okay, and on the left-hand side you have the traditional time derivative of the orientation plus terms that come convective terms that come from active stresses. Okay, so as I said, the dynamics of the polar filament, which includes orientation of the equations for local density and local velocity. Polar actin the description of the polar active filaments is slightly more complicated, but as I said, can be done. Okay. Yeah. Okay. Now, okay. So now, given that one can do these, write down these two kinds of descriptions, both of the oriented polar active fluid and the scalar version thereof, we can ask for what are the consequences. And I'd like to just describe some of the consequences of these theories. Let's see. So what happened here? Yeah, okay. So the first consequence I'd like to describe is actually taken from Vijay's, one of the organizers' paper. And this is a very beautiful example of how the scalar description of actomyosin can actually give rise to partitioning or segregation of components. Okay, so this here is the description of polarity of the cell, and the cell happens to be C. elegans embryo, and what is marked here are particular proteins called polarity proteins. They're called PAR proteins, and you see that as a function of time, you start off with a homogeneous configuration of these polarity proteins on the surface of the cell. As a function of time, you find that they partition and segregate from each other. And what they showed beautifully is that this kind of partitioning of cell membrane molecules can be understood by coupling the density of these polarity proteins to the actomyosin flows generated by the scalar theory that I talked about. Okay, so here is the equation. This is the dynamics for the polarity molecules, namely PAR2 and PAR6. Okay, and this here is a module which is like the scalar theory that I talked about, of the actin. M here is the equivalent of rho, the density of the active agents. Okay, and this here, the second equation here is just the Stokesian flow of the fluid on the surface. Okay, and so this kind of scalar description gives rise to this kind of explains very well the dynamics of this partitioning. It not only explains what they showed in their paper is that it not only explains this gross level of partitioning, but they find that the details of the spatiotemporal dynamics of the PAR proteins is well accounted for by this kind of description. Okay, so this is a paper which is referred to here, and it's a very nice and pretty example of an application of the kind of dynamics that I talked about to a real life cellular system. Okay, so that was as far as the scalar dynamics is concerned. That was one example of the consequence of a scalar theory. Okay, I'll come up with some other examples later on. Okay, here is a consequence of the polar fluid description. Okay, the scalar theory, of course, doesn't have a description of the orientation of the filaments. So you need, in order to describe the orientation of the filaments, you necessarily need to describe the active fluid as a polar active fluid. Okay, and so the second description that I talked about would be the more valid one. And what you see here is a consequence of the kind of theory that I delineated here. Okay, and this is work done by my student Kabir, and earlier done by another student, Kripa Gauri Shankar. Okay, and what you find here is a prediction of the theory, of the kind of phase diagram here, which plots in the x-axis the tendency of the filaments to align relative to each other. This is related to the number of crosslinkers that are present, which promotes orientational ordering. And on the x-axis here is the active stress. Okay, so in this phase diagram, it says that for large enough active stress, when the active stress is high and the cross-linking alignment is low, then the filaments orient to form these spiral asters or these asters, which are shown here. Okay, indeed, in a regime of parameter space, which I'm calling moving virtual defects, this theory predicts that the asters that you get have a kind of polarity. Okay, schematized here. Okay, And the prediction of this theory is that this object here, which is a localized compact object of actin and myosin, okay, with an asymmetric profile of myosin about the center, will actually move, will be motile. Okay? And this is what we call motile defects. So this theory asters or motile defects are a consequence of the fact that myosin configuration in this localized patch has a sort of asymmetry, left-right asymmetry. Okay. Okay. So now, what about do these predictions of this kind of theory bear out an experiment? And I'd like to show you not in vivo experiments, but an in vitro experiment done in Vijaykumar's lab in our group here at NCBS. There's a paper by Dariush Costa. Dariush is now at Warwick. And this is an incredibly beautiful in vitro reconstitution of the actomyosin system. As a function of reducing filament concentration, what Dariush and Kabir observed was that the configuration of actin and myosin goes from being a filament bundle to linked apolar asters. Okay, an example of which was shown, schematized by Aisha. And finally, at low enough concentration of filaments, you get these polar asters. Okay, and this is sort of shown in high resolution here. Okay, the capping protein, which is capping the plus end, shows that it's polarized. That these are polarity salted. Okay. So in fact, what happens is that in this kind of experimental system where the ATP is allowed, so this reconstitution is about actin filaments, myosin motors, and ATP reconstituted on a supported bilayer. Okay, but of course, when you put in ATP, after some time ATP hydrolyzes and runs out, and what happens is that you get these configurations which are jammed. Okay, this is not what happens in the cell, and as I said, most in vitro experiments sort of get this jammed configuration. Okay, however, what Dariush did was he had a clever way of ensuring that the ATP never runs out. Okay, there is always fresh supply of ATP, and in such cases you don't get this jammed configuration, but you get an active steady state where the asters form transiently, break up, and so on and so forth. Okay, this kind of transient steady state that comes indeed as a prediction of this theory also. Okay, because once we allow for turnover, this theory predicts that these asters will break up and reform and so on and so forth, giving rise to a non-equilibrium steady state. Okay. When we realize the situation in this kind of context, we find that that you do indeed get a non-equilibrium steady state. It's a non-equilibrium steady state where these asters form and break up, and so on and so forth. We can also quantitate this in a variety of ways by looking at both the density distribution of molecules that are bound to actin, the membrane molecules that are bound to actin, and the theory predicts that they should have non-Gaussian tails, which we recapitulate in experiments. It also, the theory also predicts that there are enormous number fluctuations of these bound molecules to actin. And again, this is recapitulated in these experiments. So indeed, both at a sort of visual, qualitative, and at a quantitative level, we realize that this configuration that obtained as a consequence of continuous ATP supply is indeed an active steady state, and it recapitulates theoretical predictions. And this can only happen if actomyosin in this system is allowed to turn over. What is the mechanism of turnover in the system? We still don't know. It could be fragmentation of filaments, as Aisha pointed out. It could be unbinding of filaments and motors. This is something that we have not been able to ascertain yet.
Madan, just a reminder about the time. We are 10 minutes to 6.
Oh, God.
Sorry.
Okay.
Yeah.
So I should... okay, so that means that I should skip a lot of this thing. So Vijay, how do you want me to do this? Do you want me to skip a lot of the consequences and of the fluid theory and go to the elastic theory?
Vijay?
You can take a call. Maybe we should have some discussion for 5 minutes as well.
Now?
No, after your talk. There's still some questions pending. So you take a call. What you?
Okay. So I think I think I've taken too much time here. But what I wanted to say that there are hydrodynamic descriptions of the actomyosin system in its fluid context. And as I said, because of turnover, you start getting an active steady state, which is very dynamic patterning, and so on and so forth. So there are many consequences of this that can be realized in the context of the cell, and I'll just relate. I've related a few of them, namely the emergence of polarity in cells and the emergence of orientation patterns. Here, it turns out that one can also describe active membranes in conjoined with this actomyosin fluid. Again, the bottom line is that this coupled membrane actomyosin cortex, which includes turnover, gives rise to excitability and dynamic patterning at the steady state. And I'll state that as a common theme. The common theme being, as soon as you include turnover, you start getting into a dynamical regime which cannot be easily arrived at by existing in vitro systems. Okay, and so both excitability and dynamic patterning happens as a consequence of the turnover of components. So that was as far as the fluid description was concerned. I'd now like to talk about, at this stage, maybe I can ask if there are any questions. I can take that and then go to the elastic description.
Okay, so there are a number of questions. Amir, would you like to go ahead now or go back? Yeah, go ahead.
Okay, if it's only 10 minutes, then maybe I should ask questions because I don't want to. It's okay if I don't finish this elastic part.
Yeah, actually, I had a general question, like from the first part of the talk, where you were showing filament bundling and crosslinks. So I wanted to ask first, like, is there any correlation between the locations of these crosslinks between these actin filaments, and and they are also linear and branch. So what governs, like, whether they will form a linear structure and what factors? So I was
right. As Haisha said, there are many different kinds of crosslinkers. There are some crosslinkers which bind actin filaments parallel to them, some in branching, and so on and so forth. And there is no understanding about what controls the spatial distribution and temporal distribution of crosslinkers. So,
okay.
Yeah. The cell some manages to regulate that.
Next question from Maitri. Maitri, would you like to go ahead and ask?
Yeah, I had a quick question. So can you just clarify, Madan, you described 3 kinds of asters. One was sort of inside pointing, the other was outside pointing, and the third was a motile aster. I assume that between the inside pointing and the motile, you're talking about an aster that's not symmetrical, but is itself sort of skewed and polarized.
So the motile asters are where they don't point radially inwards.
Right.
Projection of the orientation in some direction.
Right. And so what sort of regimes give you an outside pointing aster?
So typically you don't see that.
You don't see that.
Yeah, they're unstable and they, so you don't see the outer pointing asters. So unless it's, so typically you form these inward pointing asters.
Right.
There is a question from Vaibhav as well. Vaibhav?
Madan, hi. Does the active stress that is there, in equation, it is always conservative. It is always written as the gradient of a scalar. Is that a requirement? Has the active stress always have to be like the gradient of a potential?
No. So the force is the divergence of a stress, and the stress is a
Okay, it's a divergence of a stress. Okay. I felt it was a divergence of a potential. No, it was a gradient of a potential. It was not a gradient of a potential. Okay, I'm sorry. Yeah. And the final question I had was like a lot of the parameter, like if you're looking at the diffusion constant, etc., in the equations that you had, it had a functional dependence on various densities. The thing is, does the functional dependence of the densities there lead to a different phenomenology altogether as far as all the effects that you're showing? Or is this like assuming particular forms for these functional dependences of the diffusion coefficients or other quantities there in your equations?
Yeah, so for the dilute limit doesn't, but I mean, as in any system, when the density is higher, the diffusion coefficient would depend on the local density.
Yeah, I understand. But so like the effects that you were showing in your other works, right? They were basically in the dilute limit, or they were in the even in the concentrated limit, and there you all assumed some specific form for these functions.
No, they're in the dilute limit.
Oh, these are all dilute limits. Okay, thanks.
Yeah, so maybe I should go to the elastic description.
Yeah.
Okay. So far I've been discussing the elastic description, and again here what I want. So far I've been discussing actomyosin as a fluid, and I want to describe actomyosin as an elastic medium. Okay, and this happens when the crosslinker density is high, or the turnover of the crosslinkers, and the time scale of turnover is very large. Okay, so they don't turn over fast. Okay, and sort of simulation pictures, are like so that you see here. So again, it turns out that to see the dynamic evolution of such configurations in the cell, you require turnover. Okay, the moment so any description which does not include turnover immediately gives you a jammed configuration. Okay, and that's again the take-home message here, that you need to have turnover. So how do we put that? And this is some work that my student Dev Shankar had done in collaboration with Thomas Lekvy's group, and this is to try to describe the dense collection of actomyosin with crosslinkers in cells belonging to a tissue. Schematized here, okay, which shows a dense collection of filaments and myosin motors with crosslinkers, and also held to the cell boundaries by cadherin molecules. Okay, so the idea is to describe the dynamics of this system. Myosin binds and unbinds, and when it binds, it locally compresses, gives rise to a local contractile compression of the meshwork. But of course, eventually it unbinds and releases that compression. And what one would like to do is to describe the hydrodynamics of this collection of bound myosin bound to the meshwork. Okay, it turns out that because everything is turning over, there's a whole regime of dynamical transitions at low enough timescales. That is timescales beyond the unbinding, beyond the turnover of actin and myosin, which happens at around 20 seconds. This meshwork can be described, is a connected meshwork, and it can be described as an elastic mesh. Okay. Despite that, what one sees is local speckles such as these. If I play this, I'm not sure if it moves. No. Okay. Okay, you get little speckles such as these which move in this meshwork. Okay, so in fact you get moving speckles of actin and myosin embedded in this meshwork, elastic meshwork that you see. At much larger time scale, this whole system fluidizes because actin and the crosslinkers turn over, and the system fluidizes, giving rise to large scale flows of actomyosin enriched regions. Okay, so this is the sort of time scale hierarchy that you see. At low enough time scales, around 20 seconds, behaves like an elastic mesh. At higher time scales, the mesh gets frayed because of myosin accumulating in the meshwork and tearing off the filaments of the active mesh, and it fluidizes the system and gives rise to flows. Okay, so that's the picture that I'd like you to take away. So now, how do we describe this? Now, in addition to the kind of hydrodynamic variables that I talked about earlier, namely density of bound myosin, concentration of filaments, orientation of filaments, and so on and so forth, there's an additional feature here, and that is the dynamics of the local strain or the displacement of the actin mesh. Okay, because it's an elastic mesh, the deformations of the mesh can be parameterized by this displacement field, and spatial variations of the displacement field are equal to the strain. Okay? So this hydrodynamic description of actomyosin as an elastomer necessitates dynamics, writing down dynamical equations not only for the concentration of myosin and actin and orientation of actin, but also the local displacement of the mesh. Okay? And indeed, this can be done, and this is done this has been done by us and other people. Given here is a partial list, and I won't, again, in the interest of time, I won't describe these equations, but I want to highlight one aspect here, and I'm just going to highlight that the phase diagram. Okay, so what I'm going to highlight is the following phase diagram. On the x-axis I'm plotting the active stresses, which is related to the density of bound myosin. Okay, more the bound myosin, the more you go along this axis. Okay, on the y-axis is the local elastic stress, which is represented by the density of crosslinkers. Higher the cross-linking density, higher is the elastic modulus, B, and so you go up this axis. Okay? So this is the phase diagram that you get as a result of solving or analyzing the dynamics that I talked about earlier, the dynamics of the strain and the dynamics of the myosin and actin densities. Okay? It says that for if the meshwork is very, very stiff and the contractile activity is very low, then the meshwork is stable. Okay, so what happens is that myosin binds, it locally deforms the mesh distorts a little, myosin then unbinds, and the whole system is stable. Okay, that's this region. Okay. Now, if I put more myosin, so I move along this axis, then I move along this direction. Okay, and what happens now is that myosin now there's a higher amount of myosin. It binds to the meshwork. It distorts the meshwork quite significantly. That then induces an unbinding of the myosin, because it's a stress-dependent unbinding. Okay, and that then releases the mesh again. Then again, whereupon myosin again binds and again releases, and therefore this gives rise to oscillations that is seen in this middle region. Okay, so the oscillations that you see here are a consequence of the elastic interplay between elasticity of the mesh and the binding, unbinding of myosin, which applies contractile stresses. Okay, at very large myosin density, the whole thing collapses, because myosin, there's so much myosin, it binds to the mesh, especially when the crosslinker density is low, so the mesh stiffness is low. Myosin just tears apart the mesh, and therefore gives rise to a contractile collapse, a collapse of the whole system, as you saw in some of the movies shown by Aisha. Okay, so what I've described is in words what the mathematics tells you. Okay, I won't go through the maths, unfortunately, because of lack of time. But what I wanted to highlight is in between. In between, as I said, in one region you get oscillations, which is a result of the turnover, the interplay between turnover, active stresses, and elasticity. But in this region of this gray region here, you get a very interesting phase, and that phase is an excitable medium. Okay, it's a phase which is characterized by a traveling pulse. That is, and unfortunately, I don't have the movie here. But if I were to play, if this movie had played, then you would see that a local collection of myosin, which binds to actin, okay, forms this asymmetric profile, very much like the motile asters that I talked to you about, forms this asymmetric profile, and having once formed, it moves, this deformation moves through the elastic mesh. Okay? This is a nonlinear excitable phenomenon. It happens because of the interplay between elasticity and active stresses and significantly the turnover. Okay? So as a consequence of these two opposing features, active stresses that likes to compress or likes to apply local stresses, and the turnover which relaxes the local stresses, you get into a nonlinear regime of excitability, which is characterized, the excitability here is characterized by the appearance of these traveling pulses. And indeed, this is what we see in experiments done with Thomas Lekpi. By analyzing the speckles formed by this actomyosin within these cells, as shown in these pictures of these movies I've played. Okay, in these pictures, you see that this whole thing sort of travels and moves towards one edge of the cell, to the cell junction. Okay. Again, I want to repeat that this excitability and characterized by a traveling pulse is a consequence of turnover and activity. Okay. And it's a very and that's the crucial. Those are the crucial features that that drive it to that phase. Okay, I'll have to skip all this and try to yeah. How am I doing with time now?
This is 7 minutes past 6. So
okay.
I mean, yeah.
Okay, so maybe what I'll do is I'll talk about this. Some reason. So far, what I've been talking about is writing down a hydrodynamic theory for the elastic mesh, which is compressed locally by myosin motors. Okay? The whole system is undergoing turnover, and what we did is to write down a hydrodynamic description of this. Now, clearly, when the filament turnover becomes very high, okay, or when the crosslinkers sort of unbinding because of the stresses that are imposed by bound myosin, then no longer an elastic description will hold. The system is going to fluidize. Okay, so in the extreme limit of where the crosslinker density has reduced so much because of this of the turnover, or the meshwork has become frayed, you know you're going from an elastic description on one end to the fluid description on the other. But is there some way of analyzing the in between? Is there some other interesting new phases that are driven by this elastic to fluid transition as a function of myosin binding and turnover? Okay. And so at the moment we're trying to see if we can describe this in between regime in using a hydrodynamic description. This turns out to be quite difficult. And so what we have started now is to try to do an agent-based simulation. Okay, a Brownian dynamics simulation done by Raja Paul and his student Saptarshi Chatterjee in collaboration with my group here, where we use an agent-based simulation to simulate actin, myosin filaments which bind and unbind, and everything is turning over. Actin is turning over and myosin is turning over. And I'll just play you this this one movie that we have. The green are, of course, myosin, the red are actin filaments. And this just shows you a movie. We need to, of course, analyze these things now, but at the moment, I'll just show you this kind of movie where you can see that the myosin is sort of applying stresses, and in a stress-dependent manner, what is happening is that there's a turnover of actin filaments. Okay, so actin filaments are not only lost, as a function of the local internal stresses due to myosin, but they can come on again because of Rho signaling. So this is what turnover means. Turnover is you not only lose filaments, but you also regain filaments, happening presumably because of the Rho signaling that I talked about. So this is a new sort of area of research that we're trying to sort of go after. Hopefully, this kind of by coarse graining such agent-based simulations, we'll be able to numerically compute local active stresses, local flows, and so on and so forth. And this will give us some hints to try to construct a more hydrodynamic theory for this frayed actomyosin system, actomyosin meshwork. Okay, so this is work currently in progress, and I think I'll stop there and not talk about the rest. I'll just mention what it is, and if there are questions, then I can take it up. What I was going to talk about next was what happens if you have a collection of actomyosin and microtubule kinesin, namely a collection of contractile stresslets and extensile stresslets. What are the kind of patterns, force patterns that it can get? What happens as a consequence of turnover? Do you get interesting features? And indeed, again, you get excitability and singular force configurations. Next, I was going to talk about looking at a description of the elastic properties of a tissue. In particular, this phenomena called posterior midgut invagination, which is being shown here. It's a work done with Thomas Ekpe's group. And what you see is an epithelial tissue which is undergoing deformation because of local enrichment due to signaling of myosin. Okay, and so myosin locally compresses only those regions of those cells, and because the cells are connected to each other, it gives rise to longer wavelength elastic deformation of this tissue. How to describe the dynamics of the geometry of this tissue is what I had done with Richard Morris, who was a postdoc here and who's currently at EMBL in Sydney. And again, this feature, this again is it's akin to this traveling pulse that I talked about, right? But here it's a traveling pulse. You can see it's a traveling pulse of deformation of the geometry. Okay, and it's this traveling pulse is again a consequence of active stresses and turnover. Okay, again the same feature, excitability characterized by traveling pulse, a consequence of the interplay between active stresses and turnover. Okay, so I'll unfortunately not be able to go through the details of this very nice study, but I want to summarize now with the following statement, that in order to understand the many aspects of actomyosin in terms of force patterning and its profound consequences in living cells and in tissues, we need to include the effects of turnover and signaling. Okay, this, as I said, this gives rise to both excitability and to singular force configurations. Excitability is a consequence of this turnover and signaling, which is a consequence of the mechanochemistry. Okay, this is important both for in vitro realizations. We need to think of more interesting in vitro realizations of the cellular and tissue context, and it's important to include in theoretical models in order to understand what is happening in cells and tissues. Okay, and so with that, I'd like to acknowledge the people who have worked with me. There's a whole bunch of students and postdocs and collaborators listed here. In red are the people whom I collaborated doing experiments, and blue are all the theorists involved. And finally, I'd like to thank of course ICTS-TIFR, the Simons Foundation for funding, and a JC Bose Fellowship for funding. And with that, I'd like to take up any further questions if there are. Okay, thank you.
Thanks, Madan, for a very nice talk. Okay, so we overshot by 15 minutes, but maybe we can take a couple of questions. Vijay, is that fine? About the time? Yeah.
Please. Yeah.
So there is a first question from Haisa. Please go ahead, Haisa.
Yeah, in one of the last slides towards the end, you were talking about the agent-based Brownian dynamic simulations of networks that include turnover.
Yes.
I was wondering how you exactly do that. And I'm also curious how incorporating turnover affects the buildup of stress.
So, okay, nothing quantitative so far, but because we haven't analyzed it, this is one of the movies that came up. But the way we do it is in these particular systems is to turn over both the entire filament. We don't do depolymerization, so we turn over the entire actin filament, and we turn over myosin. Okay. And both these are threshold dynamics. That is, when the threshold, so it's given by an exponential of a local stress. So we compute the local stresses, and their unbinding rates depend on the local this exponential.
Mm-hmm.
Yeah. So there are parameters that go with that form, that hill form, and that's how we
what is the effect of having this turnover included on the amount of stress that builds up?
So as you can realize, there are two aspects of turnover I wanted to talk about. One is unbinding, okay, unbinding or fragmentation or depolymerization. In our case, we've only looked at unbinding. We haven't put depolymerization. This kind of feature has not only been done by us, but also by this reference below.
Yes, I know. Yeah.
Paper. And in both this, it relaxes the local buildup of stresses.
Yeah.
Okay. So that's obvious. But what we have done in addition, which Roger Cam and company don't do, is to also make them rebind. Okay, because this is what is happening in the cell. In the cell, because of Rho activity, Rho and ROC activity, you get the recruitment, local recruitment of filaments and myosin. Now, we have absolutely no idea about how that exactly comes about. And so the way we've done it is just to sort of again say that that somehow there must be a sensor which likes to locally look at the stresses, and if the local stress is lower than a certain amount, it sort of recruits. But the effect of that is to constantly maintain a connectivity, okay, and prevent it from getting entirely frayed. You can see here in Roger Cam's simulations that at the end of the day, it gets enormously frayed. Okay, and they tear apart and they break up. Okay, and this is because they don't include the recruitment or recovery of filaments and myosin.
Yeah.
But I think this is an interesting feature to include. And as I said, we don't know the physics of this recruitment, and so it's at the moment we're going at it blind. But the idea is to sort of try to scour in parameter space to try to see what are the various behaviors that you could get. My feeling is, one of the things that I'd like to get is to see whether there is an intermediate configuration which has something like, so for instance, if the sort of breakup and re-recruitment has a statistics which is some power law statistics, then it gives rise to a power law elastic.
Mm-hmm.
And this is one of the things I'd like to do.
Mm-hmm. Cool.
Is there any other question? Okay, so right now it looks like there are no other questions. But if there is another question, you can also go to this Slack space and discuss there. Okay, so thanks, Madan, for a very nice talk, and thanks both the speaker, Madan and Aisha, for the very nice talks today. Now over to the organizers.
Yeah, thanks, Madan. Thanks, Aisha, for a very nice talk. So that's it for the day. We will resume tomorrow, usual time, 2 o'clock Indian Standard Time. Thanks, everybody.